So, usual disclaimer. So what is Recce about? Recce is obviously listed on the ASX. Now what we have is the first new class of antibiotic in over 30 years. Now Recce is tackling the global health problem of antibiotic resistance, bacterial mutation or superbugs. We have a broad spectrum antibiotic, and keeping it simple, what that means is we kill all types of bacteria. Unlike traditional antibiotics, which are typically single spectrum, any bacteria in the blood is bad bacteria, and we therefore don't discriminate in our attraction, bonding and capability against it. We're focused on the lead indication, which is sepsis, septicemia or blood poisoning. It's the number one most expensive condition treated in hospitals, bigger than cancer, bigger than diabetes, and in fact double the second most expensive. Increasingly, it's less and less what we say and more what the international community recognises as the potential of Recce antibiotics. We were recently awarded a prestigious legal status called a Qualified Infectious Disease status by the US FDA. That status gives us 10 years of market exclusivity that only starts post-approval, plus fast-track regulatory review for the life of the regulatory process. So we really have a continuous feedback loop with the FDA and is above and beyond our patent portfolio. We'll show you the patents in a moment. They can be challenged. No one's going to take on the US government. But also importantly we're the only company in the world with that status for sepsis. So it's very prestigious and something that greatly facilitates our development in the time ahead. This is our capital structure. So here we have Dr. Melrose. Dr. Melrose is former head of research of Johnson & Johnson Australasia of a decade. He was also executive director as well. In his retirement as a polymer chemist, Dr. Melrose invented Recce antibiotics. The company started in 2008. The compound was identified in 2010, and having identified the compound, we rapidly move it through the regulatory process. There's no research for research's sake here. It's about getting to that market as quickly as possible. We have a couple of institutional investors either side. Share price, I just checked it at about 22.5 cents, so we're growing. Market cap of a measly $25-ish million. And naturally you can see in the trajectory, and you'll see in a moment, the path forward looks encouraging for the time ahead. Cash and deposits, not a lot of cash when we last reported our quarterly. We expect imminent receipt of our R&D associated with our activities. A 1-minute video.
There have been no new classes of antibiotics on the market for over 30 years. Recce 327 has the potential to break that mould. Recce 327 is a synthetic polymer designed to target bacteria that cause infectious disease, including ones that are resistant to current antibiotics, the superbugs. Bacterial infections of the blood are deadly and affect an estimated 30 million people worldwide every year. This is where Recce 327 is designed to act. Recce 327 is designed to be administered through intravenous injection, so it can quickly spread through the bloodstream. Recce 327 is designed to adhere to the outer cell wall of the infectious bacteria through hydrophobic interactions. This weakens the cell wall, and internal pressures from within the cell cause the bacteria to burst. The body's own mechanisms can then clear the infection. Normal non-bacterial cells remain intact as they do not contain high internal pressures. Tests have shown Recce 327 is fast-acting and as a broad-spectrum antibiotic is effective against gram-positive and gram-negative bacteria, including antibiotic-resistant superbugs. Following regulatory requirements for new antibiotics, safety and efficacy tests will continue to urgently explore Recce 327's ability to address the global threat posed by superbugs. Together, we can make antibiotic resistance history. Visit recce.com.au to find out more.
So the reason we touch on that is we go from the theory of what is the challenge at a clinical level and how Recce seeks to resolve that. And the challenge is large. The challenge is affecting 30 million people worldwide every year, 1/3 of which die, and it's growing at an 8 to 13% per annum and effectively compounding by nature of no new classes of antibiotic. Number one most expensive condition, it's bigger than prostate cancer, breast cancer and HIV/AIDS combined. One key statistic we look to there is I see the US as a leading medical country, yet in the US you've got over 750,000 cases of sepsis and over 270,000 deaths. That's a 1 in 3 death rate. That's what's termed an unmet medical need and a sign for things to change. And that's what Recce is focused, and our data in a moment will strongly indicate. So why are we different? All other antibiotics are natural. They're bacterial fungi, cultivated out, and they rely on what I call a lock and key specific fit, or a singular lock and key fit. In effect, you've basically got the bacteria and the antibiotic. They come together. When that bacteria mutates, which it always has and it always will, that lock and key no longer works. Recce, being a synthetic antibiotic, is designed with first principles in mind. We're attracted to the proteinaceous layer of the bacteria, regardless of its mutated form. And particularly, no matter how many times we are exposed to that bacteria, it will continue to work today and with repeated use. And importantly, without any anaphylactic reaction, which currently 15% of patients have. We touched on, via LBT's presentation a moment ago, what is the clinical problem? And the clinical problem begins at patient level when they present to the doctor. The doctor does a blood draw. They don't know what type of antibiotic that patient has. They cultivate it out on bacterial dishes. It takes 48 hours plus. There's a layer of human error in the judgment, and they do antibiotic susceptibility tests. What bacteria for what type of antibiotic? Every hour left untreated, the likelihood of survival is decreasing by 6%. You don't have time for diagnostics, and you don't have time for human error. So what they do currently is they quickly pump in a cocktail of antibiotics in the hope that it's, say, 5 or 10 different types of bacteria rather than the hundreds of types of bacteria that it potentially could be. It's guesswork. Recce is designed to kill all bacteria. Any bacteria in the blood is bad. The quicker you can get onto that patient problem, the better the patient outcome ultimately, and with a 1 in 3 death rate, you need to move promptly. We are right at the end of the preclinical studies. We've done small species, mice and rats, rabbits and dogs, and as a broad spectrum antibiotic, we've done a huge range of different types of bacteria. This is one called MRSA, or golden staph. This is in mice, all independently done. 3 groups of 10, 10 out of 10 with Recce survived, 9 out of 10 with oxacillin. Well, obviously that's 100 in every 1,000 patients not surviving, and no treatment, no good. But the reason we chose oxacillin is the best antibiotic for MRSA. If it was E. coli, Pseudomonas aeruginosa, 100 other different bacteria, oxacillin would not work. And we want to give the clinicians the capability to get onto that clinical problem at patient presentation. This is looking at it from a preventative landscape. Preventative meaning it's getting increasingly dangerous to go into the hospital and get a basic knee operation, a shoulder surgery, or any break in the skin where bacteria can get in. Here, this is Streptococcus pyogenes, or strep throat, which then becomes septic. You've got 2 groups of mice, both given a bacterial burden. One group of mice has a very, very high bacterial burden at a 12-hour time point. The group of mice that didn't have a large bacterial burden didn't, because they had Recce flowing through their veins. The preventative drip was in the animal system, meaning that bacteria was not able to cultivate and lead to the infection. So seeking to stop infections before they ever occur, and when you see that fall in bacterial count, it's not because the animals were getting better. I've shown this to clinicians, and one actually once stopped me and said, I know what's happening. Bacteria moving from the blood to culminate in the kidneys, which is organ failure and death, typically within the first 24 hours. So no time for diagnostics. We've really expanded the typical development cycle of a new antibiotic as you assess safety and efficacy. And one of the key components of it, particularly with a polymeric antibiotic, which we naturally think of as a plastic, is what's the toxicity profile? Here is touching on that we are dose dependent. We can clearly evidently see the infusion of our antibiotic in animal blood, but importantly, the absorption of that antibiotic, and the antibiotic passing in this instance rats from the blood as 100% water soluble to either break down or, if there's bacteria, have that interaction. There's no hard bits or toxic elements that remain in the blood of animals. We choose what we want in our antibiotic. We're not reliant on what's given to us in nature. And really we expand upon that in dogs. It's very important to know and to be able to track and trace your antibiotic at a preclinical level. And you'll see in a moment we've now stepped from preclinical shortly into human clinical trials, most likely here in Australia. So this is from our patents here. You've got the standard top 5 deadliest bacteria known to man. These are designated by World Health. You've got Staph, E. coli, Pseudomonas aeruginosa, Clostridium difficile, and S. pyogenes. What's important is we kill the standard form and the superbug mutated form with exactly the same ease. We don't care if it's a superbug. You don't need any more antibiotic. It will work and continue to work with repeated use. Here is a repeated use assessment. This is from our patents, obviously. Commercial antibiotic is amoxicillin. Amoxicillin sells at about $10 billion per annum. After 8 cycles with staph, amoxicillin stops working. 2 cycles E. coli, that's pretty scary because that's quite a prevalent bacteria, and 6 cycles Pseudomonas aeruginosa. We took ours out to 25 repeat cycles, and we worked as well in the same time with the same concentration the first time as we did the 25th time. Case proven. So it's very encouraging, and certainly that's what the FDA supported in their QIDP designation. How we work? Well, basically we're attracted to the proteinaceous layer of the bacteria itself. We bind to that proteinaceous layer, and we effectively have an interaction through a physicochemical process. We don't dissolve, we don't puncture, nothing nasty. We basically cause the inner cells to seek to meet their state of equilibrium. There's 10 atmospheres of pressure inside bacteria, billions and billions of cells wanting to get out. So much like 2 magnets, we're drawing the inner cells out, and you'll see in a moment that it's bursting from within outside, not from outside heading within. And that's important, and that's how we get the specificity of killing bad bacteria and not healthy cells in the body. This is an example of it. This is E. coli, different type of bacteria again. Very buoyant, very deadly, done at University of West Australia. Here you can see it bursting. I love this one because this is where you can see it for the first time at a molecular level. Pop, 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 pop. All the bacteria is bursting. And when bacteria bursts from the inside out, you've got a redundant bacteria. And as we saw in that video, healthy cells come through, mop it up, pass it through the kidneys, no more bacteria, no more problem. I can expand upon this. Basically, we've done over 30 studies associated with the typical assessment of a new class of antibiotic. Everything has been very positive. We've locked in on a commercial level 10 years of market exclusivity with the government post-approval, and extended our patent portfolio, which you'll see in a moment, beyond just the composition of matter patents, including manufacturing and curative, but to also include preventative, dosing, intravenous, oral for Helicobacter pylori and E. coli in the upper intestine. Many would say, what about the good bacteria that exists in the lower intestine? Being 100% water soluble, it breaks down either by interaction with the bacteria itself or its solubility before it gets to the good bacteria. So we look to it as a technology, not just a single product, and we're rapidly advancing down that pathway. Manufacturing, that's not just a pretty picture. Any patient in Australia that's likely to not survive without medical intervention within near months now has permission for use of Recce 327. Particularly the hospital or Dr. David Bowers, head of department there, has that for their patients who are not responding to existing antibiotics, who are allergic to existing antibiotics. 15% of penicillin patients are, therefore are untreated. Or the toxicity burden of a multi-drug resistant infection being treated with existing antibiotics is too high. So imminently, I see very high probability of inpatient use on an efficacious level in a leading hospital, or from a traditional regulatory sense under the FDA pathway, which will be naturally disclosed in due course. That's our facility, 99% product yield, bottle costs more than the liquid that goes in it, no expensive waste. The FDA recently affirmed that our quality our chemistry and our manufacturing processes to GMP grade. We're all set for human studies. And Dr. Prendergast, New York Stock Exchange Biotech Chairman, has recently joined as our non-executive director, not because we pay him a lot, but because he, in his doctorate, is a doctorate of bacteria, study of blood and bacterial diseases. That's our type of company. Dr. David Bowers there, Chairman of the Clinical Advisory Committee. He does that on a volunteer basis. That's head of Department of Royal North Shore because he wants this for his patients that aren't responding to antibiotics like they used to. And yeah, I can expand upon that. Category A we touched on. Very pleased to be rapidly moving on the business development opportunity of addressing new hospitals for consideration of their patients. In November, we have been invited to give the opening address to the World Superbug Congress. It's the largest congress on antimicrobial resistance. We are presenting and positioned as new hope in the fight against superbugs. There hasn't been a new class in over 30 years, and we'll be talking about how synthetic antibiotic development can change the antibiotic model. So we're very much looking forward to that presentation. And really, coming around to summary, we are a human clinical company in context of being focused on sepsis, septicemia or blood poisoning, applications further for gastritis, which is GI tract, Helicobacter E. coli, and we're also, with our patent portfolio, expanding into topical applications, so skin applications, particularly for E. coli infections and drug-resistant infections, and looking to the lower-hanging fruit fallback positions as our patents continue to tick along in the time ahead. So, in short, we're right at that catalytic point, having completed the preclinical trials, TGA Special Access Scheme, and increasingly the international regulators and pharmaceutical community is welcoming us to join and present the sign of new hope in the fight against superbugs. So thank you for your time.